mitochondria, they are the owners and runners of our bodies and our cells. So what they do is when they sense that there's an intruder, intentionally they block that energy oxygen pathway in order to use the oxygen that they're absorbing into the cell to target the pathogen. The Warburg Effect is an intentional response by mitochondria to combat the pathogen.
The premier source of patient-centered information on PKD and how to reverse it. This is Felix Mueller.
Hello everybody, this is Felix, your friendly neighborhood kidney hacker, coming back again with episode number 19. So this one is going to be my conversation with Mark Lintern. A lot of you who follow this channel have heard me talk about endotoxin in relation to reversing PKD for a long time. And I've mentioned it in many of my podcasts. And you've also heard Professor Seyfried on this same podcast talking about the connections between mitochondria and cancer. And we have drawn the parallels between the cancer mitochondria connection and the PKD mitochondria connection, which are very similar. So what if I were to tell you that I'm not the only one who has connected the toxin of a pathogen, which in my case would be endotoxin, to uncontrolled growth, which I think is the most plausible explanation for what's happening in PKD. Mark Lintern, also not from a formal science background, just like myself, has discovered that the Warburg Effect that we have talked about with Professor Seyfried in the context of cancer is very likely being triggered also by a pathogen, very similar to what I've been saying about PKD for years now. When I discovered this, I was flabbergasted and I had to talk to this guy. And even better, he actually wrote a book. So of course, I read the book first. It's called Cancer Resolution by Mark Lintern, and it's about his cell suppression theory. The cell suppression theory model was actually discussed with a larger group of scientists, and they voted on the validity of the theory, and they decided that this theory has just as much merit as all the other ones that are already out there in the scientific world. And I would contend it is the most plausible one. And the fact that two guys, not from a scientific field, that have their own issue and their own disease that they focus on, come to this very similar conclusion, both focusing on a pathogen that in the end triggers a mitochondrial problem, which leads to uncontrolled growth. This fact alone makes it a lot more likely that either of us are correct. So today I'm treating you to this clash of the titans, if you will. No, but really we are very aligned, and that is why I wanted to have him on. So you can listen in today on my conversation with Mark Lintern, and we will explain how his cell suppression model of cancer works. And then we'll go in what he thinks, how it can be connected to PKD, and what I think, how it can be connected to PKD, and the huge amount of parallels. And of course, also, what kind of different approaches and different treatments we could theoretically come up with that might help with PKD, as they also might help with cancer. So without further ado, enjoy my interview with Mark Lintern. Today, I'm welcoming Mark Lintern. He's a multi-award winning author of the book Cancer Resolution, ? We'll hear more about that. He's developed a groundbreaking new theory of cancer, which builds upon the metabolic theory of cancer, which we're all very familiar with on this channel. And it's called the cell suppression theory of cancer. He's not a scientist, just like myself, which is why I'm even more interested in this fascinating new model. I actually read his book and found it so fascinating and relevant to PKD, actually, that I had to have him on. So, hi, Mark. How are you doing?
Very well, thank you. Thank you, Felix, for having me on the show.
Yeah, I'm very excited to do this. You could sort of maybe give us an introduction of how you just entered this field from nowhere, basically, and share what led you to proposing a new explanation for cancer.
Oh, I'm a graphic designer by trade. And when I was 28, I developed cancer myself. It was a skin cancer, so I caught it early, luckily, and had it removed. But at the time, I was really shocked. I thought I was healthy. So I asked the doctor and the oncologist, what mutations caused my particular cancer, just from the perspective that I wanted to, prevent it in future. And I was surprised that they couldn't tell me. So I thought, that's a bit strange, because we're given the impression that cancer is a genetic disease and that this is laid out as fact. I thought, how can it be fact when we don't really know? So I thought, okay, maybe just they aren't sure because my particular cancer, they're not, they don't understand that particular type of cancer. So I did a bit more research, and the more research I did, the more confusing it became, and there was less clarity over the underlying mechanism driving the disease. So one thing led to another. I then discovered that through that research, there were other theories I didn't realize. And of course, that then opens up a whole rabbit hole of information. I then stopped the research for a while because I got the all clear from having the surgery to remove the tumor. But then a friend of mine got cancer, ovarian cancer, and she took conventional treatment. But during that path of her diagnosis, I delved really deep into the research because I wanted to investigate these other theories and what they could offer in terms of treatment, just so that I could probably, hopefully advise my friend and help her. Unfortunately, as everyone is, it's a very scary thing to have a diagnosis, and most people will put this responsibility on looking after their health on doctors naturally because it's such a scary prospect. And it was difficult for me to present the information that I identified to my friend for that reason. You know, who am I? I'm a graphic designer. I don't know anything. I'm not a scientist. Let's go down the route that is most likely to be the one that's going to help everybody, and that's the conventional route because doctors know what they're talking about. Unfortunately, she passed away after a year, but that year had really enabled me to see things in a completely different light. And I'd learned a lot of information, and I could see certain things that were wrong with the conventional approach or contentions there. And I thought, you know what, I'll write a little book, just cataloguing the information that I'd gathered, just in case family and friends come across the disease again, because I don't want anyone to go through the same process that my friend Sam went through, and I don't want to experience that either. So this information would provide a foundation for people to go, okay, there's other avenues, there's other research, there's other evidence that support a different approach, not necessarily a different approach, but an approach that can complement conventional treatment. And then one thing led to another. It became more of a passion. And 8 years later, I ended up writing a new theory because I stumbled across, on a light bulb moment, a bit of information that kind of provides a new explanation for the Warburg Effect in cancer. This was significant because at this point, I'm realizing that the metabolic theory is probably the most accurate mainstream theory. You know, so it's highly relevant. But then my theory is an addition to that. So that's the light bulb moment. And essentially, it took me 8 years to write the theory. And then I teamed up with a cancer charity in the UK. We did an event where I presented my research to scientists in front of an audience of over 200 medical professionals. And they voted to acknowledge the significance of my theory and that this needs to be tested. And essentially, in not a peer-reviewed way, but in an established way, validate the theory for further investigation. So essentially, it's sitting on a similar level to the other theories that are currently out there. So that was nice to have that validation. And then since then, I've been working with scientists to test the theory. And I've had a lot of interest from other scientists and the public who are interested in how my theory can affect their treatment decisions working forward and the survival outcome.
That's the next level, right? From theory to practice, which is, of course, a lot more difficult and a lot more, you have a lot more responsibility in that way, right?
Absolutely. And it's, as I'm working with these scientists who are volunteering their time, by the way, which is fantastic, it's becoming apparent that it's incredibly complex. And there's a lot of nuances involved in trying to just establish a test, let alone trying to identify the actual pathogens involved, because as I'll explain in a moment, pathogens are involved in my theory, just to identify them. And there's so many nuances involved with treatment, because not all particular treatments will kill all pathogens, you know? Yeah. So there's so much involved to it. It's it's quite complex, but it can be simplified. I think that's
and I want to ask you later how that, what you're studying now, how that differs from the studies that gave you the idea in the first place, right? In PKD, it's similar because there's papers out there that suggest the involvement of a pathogen, which we'll talk about, but it hasn't been sort of followed up on. Is that a similar situation in your case?
Yeah, it hasn't. It's strange because what I've found is that we go into this thinking that science is objective, and to a certain degree it is, but it's not as objective as you think in terms of problem solving. Mm-hmm. And I think this has been the issue: is that a lot of disciplines are siloed because obviously the human body is a complex machine. But these disciplines are siloed because they're so complex and they don't talk to each other very well. So what I found was, what really happened was I because I've got a graphic design background, I probably think in a slightly different way, but because I'm not involved in the industry and my reputation isn't on the line, as is a thing that happens in science. It's all about reputation, essentially. I could ask all these different questions and I could go down different routes. And I was objectively looking at all different fields. And when I did do that, I was studying infection and immunological papers. And then they started mentioning concepts to do with cancer. And then I started to align those papers and I realized there was a connection. But I realized that the two disciplines weren't really chatting about this together, because I thought, because for instance, the paper that I will mention later on explains that infection causes a Warburg-like effect. Okay, so I'd been previously studying the Warburg effect from the metabolic theory perspective, and this phrase was literally what stopped me in my tracks. And I was like, okay, so infection can trigger this Warburg effect. I'm probably getting a little ahead of myself in this podcast at the moment, but it just seems relevant to suggest it now, because I then thought, okay, surely the cancer guys know about this. And when I tried to do the research to try and find out how infection correlates with cancer, there was no mention of this Warburg effect. So they were focusing on viruses and bacteria, but the notion was that these viruses and bacteria cause DNA mutations, and then that is what leads to cancer. But there was no mention of this relationship with the Warburg effect. So that was the light bulb moment for me. And then, like you say, the issue seems to be that disciplines don't talk to each other. And so they miss important information, or they miss, it's like this problem solving step. You have two parts to this whole process. You want to, if you're a medical student wanting to benefit cancer patients and maybe solve the disease, you'll go into it and you'll be told that the somatic mutation theory or the genetic theory is the most accurate theory. Well, not even that. Yeah. You're told that it is a genetic disease, and the other theories aren't really addressed. So they're working objectively from that, through that lens. But if cancer isn't a genetic disease, then you've got a problem. So from an objective problem-solving standpoint, or scientific standpoint, you could say, there is another aspect to this, which is you step back from everything and you go, objectively, we don't actually know what the underlying cause is. We can't assert that it's this, because if we do, we're going down one route and having tunnel vision. So we need to pull back and look at everything and reconsider that information and look at all the information. But that is not what's happening. And that is what I've done, essentially, is to have a broad analysis of the data and the disciplines.
Yeah, you're sort of the connector, right? Amalgamating what's out there. And I think that's a whole new discipline, right? So I got into the whole field because I was so interested in biohacking, right? And Dave Asprey, and he found the whole thing. And he did also exactly that, right? Pulling together information from many different fields, putting it together, because nobody's doing it. I mean, we should have people whose whole job it is to like amalgamate knowledge from different studies and like come to conclusions and to actionable advice, right? That's fed from science. But it's interesting what you say about the Warburg effect and about the whole metabolic theory of cancer, because it is kind of like a magic trick, right? They just like Seyfried comes up with this new interpretation of Otto Warburg. I even had Seyfried on the podcast, and it was an amazing podcast. But in the end, they never talk about, so what's causing it? We think we know because, it's the molecular theory. But really, what you point to is that's not step one. The question is, what is the trigger for this Warburg effect that everybody's talking about? We know what it is. It's the Warburg effect. Yeah, okay, but like why? Why is it there? What is the dominant issue with the DNA theory of cancer, and why do you think we have to pivot from that?
The idea is that cancer follows a consistent path. So you have these 10 hallmarks that are recognized that all solid tumors share. So it follows this consistent path of progression. So the somatic mutation theory suggests that a sequential series of mutations leads to all these hallmarks and the proliferation of the disease. So this consistency. But what we find in the latest research, which is only really coming out about the 2013 from the Cancer Genome Atlas project, is that these mutations are random. Not only are the mutations random, but some cancers don't have any mutations at all. Okay, so that's one big flag.
Yeah, that's interesting.
So that suggests that at the very least in those cancers that had no mutations, that something else might be causing it. So at least scientists should be going, okay, we need to go down that route. You could still assume that other cancers that do have mutations are caused by mutations, couldn't you? So there's no driver mutations in some cancers. There are driver mutations in other healthy tissue that don't actually develop into cancer. So we have these driver mutations which are supposed to cause cancer, and they don't cause cancer. And then on the other front, there's not enough driver mutations in a lot of cancers. In that we're expecting around at least 8 to 10 mutations, but there's 1 to 2. So It doesn't correlate with what we're expecting to occur. And then you have things like the DNA transfer experiments. Professor Seyfried will talk about this, where if you want to really prove, I suppose, that these particular mutations in a tumor are driving cell proliferation, a good idea would be to take that damaged, mutated DNA and place it into a healthy cell, because then that healthy cell will start growing out of control because it's being run by those mutations that you've just transferred into it. But those experiments didn't find that happening at all. So there was no abnormal proliferation. So that also suggests, well, it's another bit of evidence suggesting that it's not actually going to cause not actually causing the disease. And then when we look at Professor Paul Davies talks about this, we look at the reproducibility crisis, especially in cancer. That is the fact that we have all these studies telling us what we think is happening, drawing a conclusion. But when they've gone back in to replicate those studies, they can't replicate them, which means the studies are wrong. And the National Cancer Institute in America, which Professor Paul Davies was working with at the time, did a study of about 1 million cancer studies, and they found that up to 80% are not replicable. So these are studies that form the foundation of cancer being a genetic disease, and 80% of them cannot be replicated.
Wow.
So that's huge, really, when you think about it. So a lot of the decisions being made and the treatments being developed are based on information that's 80% inaccurate, you could say, or not even correct. So there's that as well. But really, for me, it was the randomness of the DNA mutations. I was all about, okay, from a problem-solving perspective, how can randomness cause consistency? There must be a consistent factor or a couple of consistent factors that generate this consistency of the disease. The metabolic theory is great in that sense because all cancers share abnormal metabolism. Now, there's other nuances in that I will mention a little bit later. But essentially, the Warburg effect is present in all solid tumors. I will just caveat that with prostate cancer does not present with the Warburg effect, which is why I've got a contention with the metabolic theory. It only does that later on in the process. However, the cancerous tumor develops without the Warburg effect initiating it. So there's something else going on there. However, the vast majority of cancers do present with the Warburg effect. So this is a consistency. The abnormal metabolism is a consistency that Professor Seyfried correctly highlights. So he's highlighting that this is extremely important to identify the underlying mechanism of the disease. And so that's where I recognize as well that the Warburg effect was key if you want to try and identify the origin of the disease because of this consistency.
This Warburg effect it's present in PKD as well, and it's present in, as you say, in most cancer cells. So we have this type of consistency, and then sometimes we see mutations, sometimes we don't. Now we can pivot into the main theory. What do you think is the genesis of these things?
Okay, well, first I'll start off with just one or two contentions with the metabolic theory. So as I was going through this problem-solving process, I want to be confident if I'm advising or providing information to family and friends if they get cancer. So I would want to be confident in the metabolic theory. But there are a number of contentions. And those contentions are effectively that OXPHOS is not necessarily defective. That's one of the main key explanations of the metabolic theory for the cause of cancer, is that mitochondria are defective. The OXPHOS pathway is defective. And because mitochondria don't produce energy, there's this switch to glycolysis, which is the Warburg effect, even in the presence of oxygen.
For listeners, OXPHOS is the production of energy using oxygen and substrates like glucose or ketones.
Yeah, or fat, or amino acids.
Yeah, sure.
So there's this switch because the mitochondria are defective. That's the notion. However, there are a number of contentions because a lot of scientists have pointed out that under certain nutrient scarcity situations, you can actually increase OXPHOS. If you target the OXPHOS pathway, you can actually eliminate cancer. This is specifically Professor Michael Lintern was targeting OXPHOS in cancer stem cells, and you get a reduction of the tumor, which indicates that OXPHOS is active. There's a plethora of information out there, which I've got in my book, evidence to point to mitochondria still being operational to a much greater degree than is, proposed by Professor Seyfried. And can I just say, Professor Seyfried is a hero of mine. I wouldn't be where I am if it wasn't for him. So I think he's a giant in the field. And what I've done, essentially, I've stood on the shoulders of giants. I just want to, put a shout out there to Professor Seyfried. But for me, it's a bit of a double-edged sword because I'm kind of challenging some of his theory as well. But anyway, to get back to what I'm saying is, even Professor Seyfried, as all good scientists do, acknowledge there are contentions with the metabolic theory, in that he highlights when trying to explain why the mitochondria could be faulty, he can't find the specific damage or the consistent damage that would explain that. So we're kind of in a similar territory now to the contentions with the somatic mutation theory, in that the consistent damage isn't there. So, for instance, he points out that the expected mitochondrial DNA damage, because mitochondria have their own DNA, that you would expect to be damaged to cause OXPHOS to not work, isn't present. He can't find those mutations. But then he points to the fact that mitochondria under a microscope appear abnormal. You have cardiolipin abnormalities in all mitochondria. So that's a membrane within mitochondria, which suggests that the energy system is defective. However, you have contention with, again, as Professor Seyfried points out, children who have Barth syndrome is a condition that is very similar to the energy abnormalities found in cancer. So they have this cardiolipin abnormality, the same that occurs in cancer. However, cancer is extremely rare in these children. So there's contradiction. And the other thing is, if you look at oncocytomas, these are basically benign tumors. But these benign tumors are characterized by damaged mitochondria. So they can't utilize OXPHOS. And in the studies that look at oncocytomas, they conclude that OXPHOS needs to be operational in order for cancer to progress. Now, I'm suggesting that's because mitochondria, when they're active, they can produce all the other metabolites, which allows cancer cells to be plastic in their ability to choose different fuels, which contradicts the metabolic theory in the sense that the metabolic theory is saying that no, cancer cells are not plastic. Their weakness is that they only rely on glucose and glutamine, but they can't use fatty acids and other things. My research was, I was looking at that, and there was contentions there suggesting that actually mitochondria are plastic. They're not defective, and they can utilize certain pathways that are suggested to not work. The other one would be apoptosis. So mitochondria are an imperative part of the cell death mechanism. The reason why, according to the metabolic theory, that cancer develops is because mitochondria are dysfunctional, so they cannot instigate apoptosis. That's why cells don't die when they should, and we get the growth of a tumor. However, when you introduce plant compounds, natural plant compounds, so sulforaphane or bromelain, which is in pineapples and broccoli, the cancer cell chooses to die. It instigates that apoptosis pathway through mitochondria and the p53 gene, which both are said to be damaged to the extent they can't be used, hence cancer forming. So this whole thing suggested there are a number of contentions that need to be explained. And that suggested to me that there's a suppressive mechanism going on. So I've got all this going on in the background of my mind. I'm thinking abnormal metabolism is key. The Warburg effect is key. But I feel like there's suppression going on. And then I'm going off on a tangent. I'm reading about infection and how fungi, particularly on this occasion, interact with our cells. And it's this mention of when a fungus invades a cell and a cell acknowledges this is happening, the cell switches to a Warburg-like metabolism. So that, coupled with, I'd already been thinking along the lines of this suppression mechanism going on, mitochondria seem to be suppressed, and then invading pathogens trigger the Warburg effect. That was where the light bulb moment happened for me. And I suddenly realized that could it be that the pathogen is still present in the cell, which forces the Warburg effect to be sustained for a long period of time because it's an anti-infection response? And is the pathogen sustaining itself and suppressing the mechanisms within the cell? So then I did research to try and find out if anyone in the field had looked at this, and no one had. And then I thought, well, okay, what else can these pathogens explain? What else can they be doing? Would they block this cell death mechanism to explain the suppressive mechanism I was thinking about in my head? Lo and behold, plethora of information saying, yes, intracellular pathogens sustain themselves within the cell, and they essentially hijack key mechanisms to keep the cell alive. So they block the cell death process by upregulating BCL2 as one of the mechanisms, and that essentially stops mitochondria from triggering cell death. So this now fitted into the thinking I had of mitochondria being suppressed. And when you've got those two things happening within the cell, the switch to glycolysis, this proliferative state, it's a proliferative state, as well as the cell death mechanism being blocked, you then essentially have the foundation for the growth of a neoplasm, a tumor, an abnormal growth of cells, unless that fungal pathogen is eliminated. And then I came across Dr. Navio's work. Now, Dr. Navio talks about his cell danger response model, and this is really interesting because it really ties in nicely between the metabolic theory and my theory. And it describes specifically how cells react to damage or danger. So that could be a toxin, or it could be an infection. And what the cell does, it goes through a cycle, 3 phases. And it's a cycle where it responds to the whatever damage is being incurred. Say, let's use the infection as an example. So it responds to the infection, triggering inflammation, alerting the immune system to what's going on. And in that process of responding to the infection in phase 1, what happens is mitochondria, because they're so much more than just an energy system, they're actually, I think someone mentioned to me a podcast that Dr. Chaffee was on not so long ago, and he was talking about mitochondria basically are the farmers of the cell. So the nucleus is the computer, and the mitochondria actually, there's these organelles that sense everything that's going on, and they regulate virtually the cell. They are actually the owners and runners of our bodies and our cells because they're in every cell. And I believe that too. That's the conclusion I came to separately. So they're doing more than just the energy system. They're regulating everything. So what they do is when they sense that there's an intruder, an infection, they stop the production, intentionally, they stop the production of oxygen and glucose, of energy production. They block that energy oxygen pathway in order to use the oxygen that they're absorbing into the cell to target the pathogen. So they convert the oxygen into free radicals in order to fight the pathogen. So it's a defensive response. And because the mitochondria aren't producing energy now, using oxygen and glucose, the cell backup energy system in glycolysis, the fermentation of glucose, where oxygen isn't needed, that kicks in to help supply the cell with the energy that it needs in order to fight the pathogen. And several other things happen in that process to restrict the progression of the infection. So what effectively Robert Naviaux is explaining here is that actually the Warburg effect is a intentional response by mitochondria to combat the pathogen. And what you find is in that process, if the pathogen isn't eliminated, the Warburg effect remains, which means that in these three phases, which is deal with the infection, then deal with cell repair, then shut down the mechanisms of cell repair and return to homeostasis, we're stuck between phase 1 and 2 because the battle between the pathogen and the mitochondria and the immune system that now gets involved causes chronic inflammation, which then triggers phase 2. The cell can't move all the way through to phase 3 because the pathogen is still present. It's still stuck in that first phase or between phase 1 and 2. And phase 2 is the repair phase. So the cell is stuck between trying to combat the pathogen and now trying to also repair the cell, because the cell's job is create homeostasis, bring us back to this level of health. So it's stuck between those two. So you're now stuck between this proliferative state, and it's this microorganism that is orchestrating the entire process or can explain that particular process.
And that's very similar to like how we would use hydrogen peroxide on some surface to kill off pathogens, right? That's just like to simplify it even more for listeners, right? We use the oxygen similar to something like that to have it oxidize or kill off any kind of invaders or pathogens.
Immune cells, that's one of the primary weapons of immune cell. They'll engulf a pathogen and then they'll bombard the pathogen with free radicals. You look at them as if they're like bullets.
But what I love about your model is that it's, again, another sign that the body doesn't make mistakes. This tumor it's there for a reason, and we haven't been smart enough to recognize what that reason is. But just cutting it out, it also solves the issue, maybe because we're just also removing the pathogen with it, right?
Absolutely.
We fail to recognize that. And it's so interesting because we'll get to that later, but this is why your theory spoke so much to me, because a couple of years back, I made a very similar discovery in the science that showed that there's a toxin requirement for cyst production. And I firmly believe that cysts and PKD are actually protective mechanism to like harbor some toxins that have to be stored away, because otherwise they would be damaging the cells to no end.
Absolutely.
The next question that your theory poses is every human has cancer hundreds of times per year. Small tumors develop and then they go away. To me, that says this process is actually very efficient. It works very well. We have these areas in the body where possibly, like if your theory is correct, some pathogen is invading the cells switch to this Warburg effect. We start oxidizing the invaders. We succeed, and then we're good, right? So later in your book, you also go into some of the factors that you think might be the difference between someone whose tumors resolve within a day or so and someone who has a big tumor that is not going away. What are the things that prevent this cell danger response from coming to completion? Why is this battle continuing? Why is the body not winning it?
The fungal pathogen is incredibly sophisticated. When you go through and you look at the research on infection and how these pathogens exist within the body and colonize pretty much all of our tissues, it's pretty incredible because they actually are able to modulate and listen to our immune system. It's almost like, when you listen into the police radio, they're doing that. So for instance, Candida albicans can be present within the gut, and when it gets to a certain population, it can cause chronic inflammation elsewhere in the body. So it can cause disease in the lungs.
By secreting toxins which travel through the blood, or how does it work?
It's to do with presenting exosomes, which will increase inflammatory markers. I'd have to go back at that research because that's a little bit in depth, but it's just being aware that this can happen. The other thing is how fungi are able to evade detection, And the thing I want to kind of get across is that we actually thought that tumors were sterile. We thought that organs of the body and tissue were generally sterile, that there was a microbiome in the mouth and the gut, but that was pretty much it. Even the brain, because the blood-brain barrier is quite difficult to get past. However, we're now finding even in the brain, there's a microbiome present there. So what you got to think is over time, these microorganisms are able to colonize the body, but colonize the body to different extents in different people, depending on the weakness of the immune system, the permeability of the gut, and access to particular pathogens, and whether or not the microbiome is being looked after. You need to have a diverse microbiome in order to protect you. That forms up to 80% of your immune system, the microbiome in your gut, because it's the beneficial bacteria that actually feed on the yeast that's present and eliminate them. We're learning that pathogens actually invade the body. They do so by biofilms. A lot of the time they live within these biofilms, which are incredibly difficult to eliminate. Some people will get cancer and some people will get disease more than others, largely because these pathogens might or might not be present in certain tissue. If they are, it depends on people's diet. It depends on the strength of the immune system. If we're consuming toxins on a consistent basis, you're going to be damaging tissue. You're going to be weakening the immune system on a consistent basis. If you're not consuming enough of particular nutrients, vitamin D, for instance, your immune system's not going to be functioning at its optimum. So you're creating an environment, especially with the toxic world we live in, where you're providing opportunities for pathogens that are present in a commensal form in different tissue to then take advantage of that. So the premise of my theory is essentially cancer is not necessarily an infectious disease. Like, that's a loaded word, and a lot of people think of infection as a quick infection. You get sepsis, bacterial and viral. It's a nuanced type of infection whereby the terrain of the body is damaged in a particular area of the body, possibly for repeated toxin absorption. And if there are particular pathogens and a dysbiosis in the microbiome in that tissue, so there's a weakness of the beneficial bacteria that would keep fungal pathogens at bay, which often occurs because we're consuming antibiotics at an incredible rate. The fungal pathogens present in that tissue will remove themselves from the biofilm and take advantage of the damaged tissue that is adjacent to them. And that allows them then to access and invade that tissue. And that tissue becomes weak in terms of its ability to eliminate the pathogen for a number of reasons. Iron overload is one, lactic acid production is another one, which is a result of the Warburg Effect. These two compounds essentially feed fungal pathogens and they suppress the immune cells. That's effectively it. People are susceptible to this because the integrity, the defense mechanisms within the cellular environment and the general defense mechanisms of the body, the immune system, is weakened enough to provide an opportunity for opportunistic pathogens present at the time to invade cells and establish themselves. And when they're inside the cell, they can then produce metabolites or manipulate the cell and evade immune detection. So one of those things would be an upregulation of PD-L1, which effectively is a receptor that allows those cells that upregulate PD-L1 to hide from the immune system. It's telling the immune system that we're healthy, there's nothing wrong with us. And this is something that, when you look into the literature, fungal pathogens do. Yeah. They also produce something like a metabolite called nagalase. And nagalase breaks down the messaging system that alerts macrophages to an infection or a problem. And there are many other different mechanisms that these pathogens, these very intelligent pathogens, are able to manipulate to actually evade detection. And once they're in the cell, it's very difficult to get them out, especially if your immune system is compromised.
This is how your theory explains how we can get this consistent disease result from so many different triggers that we're told that cause cancer, right? Beginning from radiation to chemicals to the sun's rays, obviously.
Even sleep deprivation.
Yeah, sleep deprivation. That's great. The immune system. But yeah, I mean, the sun's rays, I'm not sure about that one. Maybe it's also the sunscreen. This is how you explain we get this consistent disease presentation from all these different types of insults, because it'll change the environment, but the commensal biome is enough in many cases to take advantage of this weakened environment, right? So everybody has probably some amount of candida, and other pathogens. And as soon as the weakening is there is a chance of this happening. And then if the weakening is strong enough, like if the body is weak enough, then the cell danger response, like the cells trying to fight off this invader with oxygen, like oxidizing the invader, might not be enough to actually fight it off, which is then how we get the actual tumor, and then it's sort of a runaway train. Is that how you could summarize it?
Yeah, pretty much. I mean, this is one of the major things that I explain in the book, and that no one else seems to be able to explain. Why do so many features, different carcinogens, different factors that cause such random damage cause the consistency of the disease? You know, how can sunlight, how can lack of sleep? And when you track it all back, it all comes back to a number of factors, which is essentially immune weakness, the compromise of the terrain, so the increase of iron overload, that's the release of iron into the tissue, this lactic acid response, so chronic inflammation, which produces lactic acid, and then this other factor, where the pathogens are present and they can invade the cell. And they then trigger the consistency of the Warburg effect, this metabolism that we see. And so you can explain all this inconsistency, all this randomness, all the random DNA damage that occurs through the change in metabolism caused by the pathogens that are present at the time in the tissue that's damaged, and they're taking advantage of that. This also explains why certain people don't get cancer and certain people do. So someone who smokes might not get cancer, and someone who doesn't smoke does. Those pathogens might not be present in sufficient quantity at the time to actually take advantage of any damage that occurs. So you might have a really good microbiome, a really good immune system that can ward off these pathogens.
And also very interesting, I have to throw this in there. I learned this like 10 years ago. If you look at the correlation between smoking and cancer, smoking unfermented tobacco is not correlated with cancer. So the fermentation is actually what introduces fungal pathogens into the tobacco, which we then burn and inhale.
Ah, interesting. Yeah, that's interesting, because I hear Dave Asprey talking about coffee as well, and how fungi are present in the coffee that you eat if you're not having really good coffee. There's really interesting information about Tasmanian devils. So Tasmanian devils are under threat because there is a transmissible form of cancer. What happens is they bite each other. They're very aggressive. They bite each other on the face, and then they develop a tumor on the face from the bite.
Wow. Really?
Now, what we find is that the mouth is the next largest consortium of microorganisms in the body. So what effectively I would be saying is that you're with that bite, and there might be a particular type of fungal pathogen, or even bacteria involved as well. Parasites are involved as well in this. You rarely ever get an fungal infection, without bacteria, viruses, and parasites. So there is a synergy there, and when you look at the evidence, they do work together to help each other out. Obviously, the conclusion would be that the reason why these tumors are so proliferative in these animals is because you're transferring into that wound the microorganism that then triggers the Warburg effect, and then everything else after that.
I wonder if this kind of experiment has been replicated in the laboratory.
To a certain degree, there is evidence to suggest that is already happening.
I mean, we do have the cancer viruses too, right? But again, they probably just lower the immune system at that area.
Well, I see viruses as a carcinogen, in the fact that they terraform the terrain like a carcinogen would, damage the terrain.
Yeah.
They weaken the immune system, which enables the fungus to take advantage of it. Now, the interesting thing with viruses is you've got a process called vomocytosis, and the immune system prioritizes viruses over fungi, to the extent that if a macrophage is engulfed a fungal pathogen, to obviously eliminate it, if the threat of a virus is then suddenly present, the macrophage will expel the fungal pathogen wherever it is in the body and ignore the fungal pathogen, because the primary focus is the virus, because the virus is going to cause more immediate damage.
So you mean it'll expel the virus?
No. The immune cell will expel the fungal pathogen that it's. So it's like the analogy would be a police officer. You see a petty crook trying to break into a house. There's a police officer. You go grab the crook, and then you get on the radio. Oh, we've got a robbery ongoing. So you can't deal with this particular process. You need to prioritize the robbery. So you
Right.
let him go. So what the immune cell vomocytosis is the process of which immune cells expel, get rid of, or allow the fungal pathogen back into the environment in order to
Right. I
focus
see.
on the greater threat of
I
the
see.
virus. So,
Wow.
I mean, this might have implications with COVID as well. We're seeing this higher viral load and this acknowledgement of turbo cancers. So
Yeah.
there could be a relationship there.
Yeah, and we do know that the spike protein does do something to the immune system to lower it, right? And then
I've heard that too with CDT4 and CDT8 cells.
yes,
So if you've got a process of weakened immunity and
Yeah.
then the immune cells are focusing or ignoring fungal pathogens, if they're elsewhere within the body, they're just going to throw the pathogen back out there. So you can see how
Yeah.
fungi are able to colonize different areas of the body as well, just through
Yeah.
that process.
just for listeners, you could do a blood test of part of your immune system and get the CD8, CD4, and other parts of the immune system tested, and then you get sort of a report back, and you can see if you're well staffed in that area or not, and if you're not, then you know that you have an issue, and that can always lead to cancer and other immune diseases. Let's now use this model, which I find so fascinating and makes so much sense, and sort of see how you would apply it to PKD, because this spoke so much to me because I found these studies a couple years ago. This is germ-free mice. That's also germ-free mice as a model, and they all have a PKD-like mutation that causes kidney cysts. And as soon as they remain germ-free, they have like 5% kidney cysts. Like 95% are free of kidney cysts, even though they have this mutation, which supposedly is deterministic and always leads to kidney cysts. And actually in humans, it hasn't ever been described there's somebody with a mutation that doesn't have cysts. So for now, we assume, the standard science assumes that it's 100% deterministic. I don't think it is, but maybe we just haven't ever had somebody with the right conditions. Maybe we have because we just didn't look for them. So that's unclear. But we do know that germ-free mice have very good chance of having no cysts. And then also, there are experiments where they take the cyst fluid out of a PKD kidney and put it on kidney tissue of non-mutated cells, and it will also grow a cyst in that tissue. On the other hand, let me mention a third study. If you take a cyst, you excise it from a human kidney, you put it in a petri dish, let it grow, then you replace the cyst fluid inside this mutated kidney cyst that is growing the petri dish. After 72 hours, the cyst completely resorbs all the fluid and goes back to working kidney tissue, being non-functional before.
And that's even though it's got the mutation.
Yes. So without any other information, which of course we can talk about as well, what does that trigger in your brain?
Very similar to when you're looking at the somatic mutation theory and saying that mutations are the underlying mechanism driving cancer, is that you would expect the mutation being present. Even if you remove most of the cyst fluid, the cyst would then reform if that is the underlying driver. So it suggests to me that the mutation isn't the primary driving feature. There must be something else that the mutation does to facilitate something else. Clearly, the mutation facilitates the disease, like a number of mutations facilitate or increase the risk of cancer. But when you look into, you've got to look into it further and go, well, why is that? What are the other correlations? Does it facilitate something else? And that's where we always need to, I think we always need to go whenever we're trying to problem solve anything. Why? Ask again, why? Until you get to the factor where you can't explain anything else.
So the things that we know about the PKD mutations for now, or the one that I'm aware of, is that like there's like many different ones, but there's two main mutations, PKD1, PKD2. They are used to form the PC1 and PC2 proteins, which are doing two things that I'm aware of. One is they regulate the intracellular calcium levels, and the mutation lowers intracellular calcium, doesn't increase it, which some people would expect, but it lowers it. And also, they are involved in cell adhesion. And it's not entirely clear if they worsen it or not, but it hasn't been studied that much. And I assume that it's probably worsening cell adhesion. So, and we talked about this in another discussion that we had a while back. And you had a very interesting thought when you heard that thing about the cell adhesion. Maybe you can think of it again.
I did do a little bit of research on PKD before coming to you today, and specifically on E-cadherin. And as far as I'm aware, that mutation inhibits E-cadherin. So we've got a reduction in E-cadherin itself, the protein.
For listeners, let's explain E-cadherin.
So E-cadherin is like a cell adhesion protein. So it allows the cells to bond together. There's a number of other features that it performs. It's associated with cell proliferation, stemness, and it effectively works within these tight junctions that acts as like a block, so a block to infection. So it's also associated, because they're junctions that connect cells together, they're also associated with metastasis in cancer. Because what we find in cancer is that E-cadherin is downregulated in most cells.
Which means that sort of the door of the house, if the cell is the house, is not really closed that much?
Yeah, so I suppose it would be like the porthole windows of a boat or a submarine. You want to close them, don't you? But they're open, so it's going to sink. The interesting thing is, because there's a number of mutations in cancer, and always the thing will be, when I speak about my theory, is people say, well, what about these particular mutations? They increase cancer risk by 80%. When you look at those particular mutations, there's a consistency. And the consistency is that they inhibit the production of E-cadherin. Why is that? So why is that relevant? So then you kind of look down the route of, okay, what relationship does E-cadherin have to pathogens? Well, turns out that fungal pathogens actively reduce E-cadherin in order to invade the cell. It enables them to invade the cell. But also, if you have a mutation that already does that, the cell is now open and vulnerable. So it's easy to facilitate this infectious process. So arguably, the reason why the risk is increased is because the barrier to infection is now reduced, and it's easier for the pathogen to invade the cell. But then this is associated with proliferation. So you could have the proliferation of a cyst because, you've lost the control mechanism that E-cadherin would normally provide. I didn't realize, but there is that association with the PKD1 gene mutation in this disease, which is very interesting. But the other thing, so I have this suspicion now, going on what our conversations previously, and just a little bit of research on the subject, and I'm just grazing the surface, but I kind of think that there's a bacterial fungal component where the bacteria probably might be dominating. It's triggering the Warburg effect, so it's effectively present intracellularly. However, these pathogens are brilliant at hiding within that tissue. They're secreting particular toxins, as they all do, depending on what they're consuming, but also depending on whether their goal is to continue hiding from the immune system or break down certain tissue. So they're producing like an endotoxin, which you were talking about earlier, and that paper you mentioned talks about is present. The immune system is being alerted, inflammation is being triggered, the immune system is coming in and trying to deal with the toxin. Immune cells die when they deal with toxic substances as well. So, but then they, as we often get with any injury, you get pus coming out of the injury. That is essentially the immune system working to eliminate whatever it is, the problem, and dying in the process. So you have this fluid increasing, which is combined with the toxin, damaged kidney cells, and damaged immune cells, and that is encapsulated within this cyst because it's a process of trying to shut down and control the pathogens that are present. Because the pathogens aren't being eliminated by the immune system because they're clever and they've hidden within the cells, but they're still continuously producing this endotoxin, the immune system is continually arising, it's continually inflamed site, and you have this growth of the cells. But also, as an interesting characteristic, we always have to look at the metabolism of the particular tissue because it's different in particular tissues, and that's going to affect the response. So I note that in kidney cells, they have these active fluid pumps for what they're cleaning particular substances within the body. So I wonder, and I haven't gone into any detail in this, but I wonder if there's an element of the fact that these cells exchange a lot of fluid also adds to the fluid within the cyst as well. So there's going to be something involved with that. But also the kidney has a special ability to replicate when damage occurs. So they almost transfer, they de-differentiate into a stem cell-like state. So they're very good at replicating without there being stem cells present. So these are normal cells, and this happens in cancer as well, but on only at a more extreme level. So during this process, I was thinking about all of this and thinking that seems to be a good model to explain this. So if the bacteria and fungi are hiding within those cells, then I would expect PDL1 to be upregulated, which allows cells to hide from the immune system, because that's what happens in cancer that's what happens with fungi in cells. Lo and behold, PDL1 is also upregulated in PKD, which again, so all this is kind of suggesting there is this involvement of bacteria and fungi that is triggering it. I kind of feel like the fulcrum is this constant production of a particular endotoxin, which is drawing these immune cells to the site of damage, which then facilitates the growth of the fluid within the cyst itself.
And the question is it in the gut, or is it actually locally in the kidney?
That's an interesting question. It could be both. I initially am thinking because it's localized to particular areas of the kidney, probably within the kidney.
But it's also, I mean, many people's in the liver, some had cysts in the spleen, but it's always in areas, you always get the cysts in areas, as you say, that already have tissue that is involved in secretion. It can even be in the testicular area where that fluid is produced. So it's always related to fluid production in some way.
So I didn't realize it went to the liver as well. Does it progress later on to the liver? Is that what you're saying?
It doesn't have to do that like in a chronological way, but most have them, some don't.
The question then would be, if it is extracellular and it's coming from the microbiome, so what you're suggesting is that the endotoxin is being produced somehow. I suppose it's being filtered through the kidney, but it would be filtered through would it be filtered through the liver first, being as though the liver is one of the primary toxin removal organs? I would expect probably there would be an issue with the kidney, but then you've got a lot of stem cells in the kidney, so maybe it's able to not affect the kidney so much.
And it doesn't have to be just endotoxin LPS. We don't know that for 100%. It looks like endotoxin LPS does cause that, but maybe there's also something else that does the same thing. So, because, I mean, the specific studies just haven't been exhaustively done for so many different potential pathogens, right? But we do know that there are studies where they use like NDGA to injure kidney cells, and they can injure them, but they don't get cysts even if they have the mutation. But as soon as they add LPS to the solution, then they will get a cyst with the same tissue. So it definitely looks like LPS is involved, but it might be other things as well.
Yeah, I think the fundamental other thing is that it depends on the pathogen. There are so many different pathogens, and they will react and operate in very different ways. So I'm saying that for cancer, it's a particular type of fungal pathogen or combination of bacterial and fungal pathogens that work in synergy. And you're going to get, maybe it could be dominant with bacteria, with PKD. Because they're operating in a particular way, that's what forms the cyst. But also the tissue itself, it's going to be because of the functionality of that particular tissue.
How would you come up with, what would be your best study design idea to test the theory? Testing the involvement of a pathogen in PKD. I have some thoughts, but I'm interested to hear like how you would go about testing this idea.
Initially, if I thought it was a bacterial, it would be obviously antibacterial drugs. Would be the first shot.
Would that help if endotoxin is already preformed and present?
It depends. Is the toxin contained within the cyst and it cannot be removed, and that's the problem? Or is it that the toxin is being continually produced? If it's being continually produced, then that suggests that the pathogen is present. So if you eliminate the pathogen, what you would potentially get is a remission, in the sense that the cyst wouldn't grow anymore, because you're not producing any more of the toxin and bringing more immune cells to the site as much as you would be. And then you would look at detoxifying and looking at strategies to support the kidney tissue in reducing the size of the cyst itself. I'm not a clinician, so I don't understand particularly those principles. But maybe also because of the Warburg effect, the benefit appears to be that the pathogens present also can consume primarily glucose. The Warburg effect will help aid that process. So you might want to use ketosis.
Sure, and we are,
yeah, because ketosis enables or triggers and facilitates mitochondrial biogenesis, so you can improve mitochondrial function, which again would be the defense mechanism against the pathogen. So yeah, a combination of initially trying those. Is antibacterial drugs, has that been tried with PKD?
Well, what I do know is that if they do like organoid research or petri dish, they always add antibiotics to the solution, just as a standard precautionary practice.
To eliminate contamination.
Yeah, and the researchers that I talk to that do that, they're even convinced that it couldn't possibly be some kind of bacterial involvement, because they're convinced that there can't be any bacteria in the solution. But then you look at the effects of endotoxin, which could even it can just be contaminated with it. As soon as the endotoxin is there, you don't have to reproduce it. If you look at what endotoxin does to cells, it's inverted when it's a non-conditioned medium, and it's connected to ceramides and sphingolipids, and I haven't really dug into that deep enough.
So when those scientists are working with in the petri dish and they're applying the antibacterial drug, does this cyst still grow?
Yeah.
And so you're saying that could be that's down to the endotoxin that's potentially present.
Well, it might be that that's already there because it's hard to avoid. I mean, there's even, it's a big problem in any kind of raw materials that they get for these kind of experiments. And I guess the thing that could be tried is adding something that's binding endotoxin, like polymyxin B, for example, can bind endotoxin, but it's very, very cell injuring in itself.
Well, then you've got to ask, is there a fungal component?
Exactly.
The study that I looked at and the study that you just quoted a moment ago identifies polysaccharides from fungi being present within the cyst. So, and rarely is there ever, like I say, when you get infections of particular pathogens, it's bacteria and fungi and viruses as well working together. That's definitely something that needs to be investigated too. So again, I wonder, would antifungal drugs ever?
And antibiotics, they could even increase fungal growth, right?
Well, yeah. So I've just recently looked at a number of studies which show that the risk of cancer increases in a dose-dependent manner with antibacterial drugs. So this is one of the reasons why I focused on the fungal pathogens.
And they are made from fungus, right? Antibiotics.
This is it. The whole process of me doing this investigation got me down to thinking on the microorganism level, on the cellular level, trying to actually get into the minds. It sounds a bit crazy, but trying to get into the mind of bacteria and these infectious microorganisms, because they are intelligent. We've seen recent studies showing how fungi can move around and avoid certain objects within their environment. And the mycelium they produce, it's like the synapses of a brain. It's quite incredible. They are actually a lot more intelligent than we give them credit for.
It might not be endotoxin. It might be other polysaccharides that originate from a fungus as well.
Yeah. So we do need to be looking at that antifungal aspect of things.
It could always be both. For a long time, I thought, it has to originate from the gut. Of course, it could also be that there's already colonization of some pathogens in the actual organ itself. But even with the gut aspect to this, another interesting thing to look at is transplantation, right? Because if we assume that it's just about the pathogen and that the pathogen is sort of the issue, then we would expect cysts to grow in transplants. But they don't. I'm not sure if there's very, very, very few exceptions where it sometimes has been seen.
You mean that if it's in a transplant, if it's just genetic, that we expect it to grow?
If you're going to the other extreme and we're saying the PKD mutation sort of leads to leaky tight junctions in the gut, so we get more pathogens in the bloodstream, which then have to be filtered by the kidney, then that would also affect the transplanted kidney.
Right.
But it doesn't. So I think that's another thing that points towards what you were saying, which is the tight junctions being weakened, which then sort of gives us the 1-2 punch, which is maybe a heightened level of pathogens in the blood, and then also the inability to get rid of them properly because E-cadherin is downregulated and it gets into the cells where it shouldn't be going.
Yes, so a transplanted kidney, you wouldn't have the mutation.
Yeah.
And then even though you've got the pathogens present, it's much harder for the So the terrain of the new kidney is not damaged to the extent that it would benefit the pathogen and enable it to infect, which is the same I'm suggesting is cancer. If the terrain's not damaged and even if you have the pathogen present, you're not going to get cancer. It needs to be an opportunistic moment where it benefits the pathogen, allows it to establish itself. So you're not going to have that.
Do you know if in cancer there's any receptor or things like that you can genetically alter or remove or block to inhibit the cell danger response? For example, if you add endotoxin to any kind of culture and you inhibit TLR4 or you remove TLR4 genetically, then there's no reaction to the presence of the endotoxin, which is one of the things that I thought about could maybe be pretty simply be done in a petri dish or organoid model. Just remove TLR4 and see if we still get cyst growth or if we can trigger it with adding extra pathogens.
Yeah, that would be interesting. So we're trying to determine whether or not it's the actual reaction of the cell that's the problem or the pathogen itself.
Is there something like that in cancer where you can alter some kind of receptor to the pathogen so that it doesn't have the same effect, doesn't trigger the cell danger response? Is something like that known?
I can't think off the top of my head at the moment, but I'm absolutely sure there will be. Well, we do have toll-like receptor upregulation in cancer, the early stage of cancer. I think TLR4, maybe TLR2. When you look at those particular receptors, they are associated with infection. We have this. So when we're looking at the beginning process of cancer development, we see this upregulation in these receptors that are designed to identify pathogens, and that suggests that pathogens are present in the very early stages.
Some other things that we've found helpful in PKD that have been studied and that we are applying. So ketosis works pretty well. There's been studies doing that in animals, and we have done it in humans for almost 10 years now, and we're seeing cyst growth reduction, some actual reversal of cyst growth. So we're going back, like some people go back like 10, 15, 20% in kidney volume. So it seems that some of the cyst fluid gets expelled or filtered or whatever under the influence of ketosis and fasting. Fasting is very efficient in lowering cyst volume, like even in a matter of days, actually. If you do like 3-day fasting, in mice at least, you'll get measurable cyst volume reductions and also kidney volume reductions. And then also things like quercetin, right, which also you talk about in your book, like the bioflavonoids, the polyphenols. Quercetin is very effective in some rat studies to, it like reduces cyst growth. It's something that we give almost to everybody. Curcumin, ginkgo biloba, these things seem to do a good job at reducing cyst growth together with ketosis. So, in your mind, does that also fit with the whole pathogen idea?
Absolutely, because what we're looking at is strategies that are actually directly and indirectly antifungal, antipathogenic. So studies into ketosis show that you can hugely reduce the pathogen load within the body through ketosis, primarily because you're restricting glucose. And this seems to be consistent throughout a lot of diseases within the body, because these pathogens, a lot of them, not all of them, are their primarily fuel is glucose. So whenever you have high glucose diets, we see disease occurring. And a lot of this is related, I think, to the pathogens that feed on it.
So you're saying ketosis works not because we sort of inhibit the Warburg effect, but because the fungus can't use the ketones, or that would be one idea.
Yes, I've noticed that ketosis doesn't really work in cancers that obviously are not dominant with the Warburg effect. Now, my argument with this is that those particular cancers appear to be influenced by fungi that are lipid based. They feed on fats. So Malassezia, for instance, and that's related to cancers like pancreatic cancer, which isn't triggered by the Warburg effect, which is interesting. So that would be the explanation for that. But no, ketosis works in the sense that it's what you're doing, and just the very fact that when they study patients, infection is reduced on a massive scale. But ketosis reduces the pathogen burden within the body because those pathogens primarily, apart from Malassezia fungi and those particular fungi, they rely on that food source in order to become virulent and strong, because a lot of the cellular structure of the body of the fungus is primarily polysaccharides, so glucose or carbohydrate. So that's going to be one major feature. But ketosis also generates a catabolic state within the body. So the body's going to get rid of damaged mitochondria, damaged cells, and regenerate itself. So you're not only you're improving and regenerating immune cells, which is why ketosis and fasting can be beneficial for cancer patients undergoing chemotherapy, because they have reduced side effects because the immune system is stronger. But you're also increasing a mitochondrial biogenesis. So you're improving mitochondria. What I also found was that in cancer cells, and I think in PKD as well, you have this process where you can increase the efficiency of mitochondria from adjacent cells. So you have this horizontal transfer of whole mitochondria that come into cells that actually increase the benefit of those cells. So in effect, with ketosis, you are going to improve the operational ability of the mitochondria, which then will improve its ability to defend itself against the pathogen or take back control of the mechanisms that the pathogen was controlling. And at the same time, if you're reducing glucose, and that's the primary food source that is being absorbed into the cell because of the Warburg effect, essentially that increases the glucose absorption rate of 18 times, then you are going to be feeding the pathogen, and you're going to be restricting the pathogen with ketosis. So in that environment where the pathogen needs to stay strong to sustain its production of antioxidants and what have you, it's going to be weakened, and the mitochondria are going to be improved in their ability to take control back from the pathogen.
Well, if you actually consider different pathogens being able to all produce their own toxins, and maybe even sometimes the same toxin, even though it's a different pathogen, and that's a very interesting way to explain like differing rates of response to ketosis or to other things that we give, like quercetin or curcumin or whatever, because the toxin might be the same, but the pathogen that produces the toxin might not be the same. So we might have to sort of tailor the approach to whatever pathogen is there, which of course can become very difficult because we don't even know where to look.
Like I say, if Malassezia is present as well as part of the microbiome, then it's not going to be affected by ketosis because it feeds on fat. The other aspect of the question you were asking there is quercetin and all these other compounds found in plants. That was another thing that I wanted to explain, because it's not explained in cancer circles specifically. Why do these natural compounds, why are they able to kill cancer stem cells, for instance? Or why does the cancer cell decide to kill itself when in the presence of these compounds? Well, that's something that hasn't been explained yet. And it just contradicts the mainstream perspective, because the mainstream perspective is that malfunction to the cell leads the cell to lose its control mechanisms in the surrounding tissue. So it develops a mind of its own. So it wants to survive by itself. So it chooses not to die effectively. That's the suggestion. And yet, when you drop these natural plant-based compounds onto cancer cells in a petri dish and healthy cells, the cancer cells selectively choose to die, which is the opposite of what we're being told. Yes. Why would that be? So how can we explain this? Well, for me, those compounds, when you look at them, they're highly antimicrobial, highly antifungal, highly antibacterial. That would explain the reason why those compounds are so effective, especially with cancer stem cells. And the other point that correlates with this, which highlights this or emphasizes it even more, is when you look at honey and silver. So honey is effectively primarily sugar, which should essentially feed cancer and possibly PKD. However, when you do the same thing in a petri dish with honey to cancer cells, they commit cell death. So why is this substance that should be feeding them killing them? Because, well, honey is highly antimicrobial. We use it in wound healing for that very reason. And then again with silver. So silver is interesting because it's not even a nutrient that the body utilizes. Okay, so the body can't benefit from it in a nutritional sense. Yet when you introduce silver to cancer cells, they also choose to die. Why would that be? We all know that silver is highly antimicrobial. And it can cause DNA damage at extreme levels, but the studies I was looking at, the healthy cells do not die. So the level of DNA damage is very, very low. And effectively, if cancer cells can't die because they're not expressive to DNA damage, they're more hardy than healthy cells. What you would see if DNA damage is the mode of action is that the silver would be toxic to the healthy cells and they would die first over the cancer cells. But that's not the case. So we know that it's not the DNA damage that's causing the elimination of the cancer cells. It must be the only other factor that I can see is the antimicrobial properties. And that then again highlights that there's this link potentially to microorganisms driving the disease. If you eliminate the microorganism, you give control back to the mitochondria, that they can finish the cell danger response. When they get to the end of that same cell danger response, what they do is they look at the cell and they go, we're damaged beyond repair. We're damaged to the extent that we're a danger to the host. So we'll enact our programmed cell death mechanism, cell suicide. Yeah. That's what our cells are all programmed to do. And then they die. So.
Yeah. And we are seeing actually, maybe in 10 to 20% of PKD patients, we're seeing like 10, 15% drops in blood pressure when they take silver. Which is not described in the literature at all. Like if you look for silver and blood pressure, almost no results. Just anecdotal from like 3 different forums that you can find online. And I was thinking maybe that's a secondary issue because not all PKD patients have high blood pressure, but most of them do. But yeah, it looks like there's some pathogenic component to high blood pressure as well. And it fits with the whole idea. What are some approaches that you've recommended to people to sort of start normalizing the biome, be it in the gut or maybe also transmitting to other organs that may have their own biome?
What recommendations I would have for cancer patients effectively? Is that what
Sure. I mean, I assume that it's going to be very similar in PKD because we're looking at pathogenic organisms.
Yeah, I think so. So it would essentially be address the terrain first. I mean, from my perspective, I'm trying to uniquely explain cancer from a theory perspective because that's science-based. Treatments are developed from theory. So you've got another theory called the tissue organization field theory, which suggests that cancer is a result of tissue disorganization. So damage to the tissue causes cells that normally operate together and communicate together to no longer communicate. So the barriers to proliferation have been broken. So the notion being that the underlying state of any cell is proliferation. Like a microorganism, the underlying state is proliferation. Yeah. And the point of my theory highlights that actually, yes, the terrain is damaged, and that then allows, rather than the actual cell growing just out of control because the signals aren't there, it allows the fungal pathogen to come in. So that's one of the major problems. So address the terrain first, because it's quite clear through that theory that when you do address the terrain, you can reduce the cancer. So what they've done in their studies is they've taken cancer tissue, I think breast tissue, and they put that cancer cells within healthy cells, and those cancer cells return. Similar to how you were saying with your experience when you flush the cyst out, the fluid, they return back to normally functioning breast cells.
Yeah.
Now, I would argue in that situation, that's because the terrain has changed. And so you have a terrain now which is now hostile to the fungal pathogen. Possibly the terrain has got a beneficial microbiome that would help to eliminate the pathogen. It's no longer in that environment that is conducive to the pathogen to being able to sustain itself. And therefore, you get this return back to normal function. So the first thing would be to eliminate infection, eliminate toxins, look after the terrain, do everything you can to bring the terrain back to homeostasis. And I'm assuming that would be the same for PKD.
Yeah.
And in that, it's specifically addressed the microbiome. And then on top of that, it's also addressed the immune system. A lot of times with inflammatory diseases, the immune system is actually quite weak. One thing which will quickly point, I'll come back to this in a moment, but I'll mention glutamine right now. I'll come back to that in just a moment. So it's addressed the terrain, the microbiome, the immune system, because that's paramount with everything, obviously. And then it's to address the abnormal metabolism. You want to assume that the metabolism is bad. That could be the underlying mechanism. We know that's changed, so address that, ketosis and what have you. We discussed that already, because you want to improve metabolic function. Even you might want to consider, we're talking about ketosis, upregulating mitochondria. You might want to consider something like methylene blue, a dye that actually increases mitochondrial biogenesis as well as enables the increase of OXPHOS and the increase of reactive oxygen species produced by mitochondria. But also the interesting thing is methylene blue is antimicrobial. So it's antifungal. And it works on a number of levels on the fungal pathogen. So I hear that there's benefit in the cancer space.
And it's also great for UTI. It used to be prescribed for
yes,
that.
Yes, it did, yes.
And that's a big problem in PKD patients, which is another hint.
Yes, absolutely. So there's all these other different aspects, but the fundamental thing is the metabolism, supporting the mitochondria if you can, so changing the acidity of the cell, because the Warburg Effect essentially generates a lot of lactic acid.
Sure, and we recommend that to patients to monitor their second morning urine pH and use things like magnesium citrate and, if needed, sodium bicarbonate vegetables to get their pH into the optimal range with very frequent home testing until they sort of get into the zone.
Absolutely. That's pretty much the main bulk of what everyone needs to do and what people are doing in cancer when they're looking from the metabolic standpoint. They are technically doing that. They're addressing the terrain, the microbiome, the immune system, and abnormal metabolism.
What would you do concretely to address the terrain? So are we talking get a gut microbiome test and then sort of do antifungals if needed, take probiotics? Do you have any specific things that people are actually doing?
Well, that's a good question. It's a number of things. It depends on your microbiome and how you deal with certain nutrients, because we don't all react in the same way. I have a couple of problems with absorbing, say, some nutrients, like cheese and butter and things like that. I don't have anything specific to say, do this, do that, because everyone is different. But from that perspective, it is just this notion of eating organic-based produce, primarily plant-based, to the extent that you've got all these antimicrobial compounds that are present in those, but also some meat, primarily because it's more bioavailable to get certain nutrients, and then also utilize certain supplements. Don't go overboard with the supplements, but you need to make sure you're hitting the optimum level in order to make the immune system function optimally. But also you need herbs that can chelate certain toxins out of the body, because the whole idea is to reduce the inflammation.
Yeah, I think maybe for a minute or so, you can talk a little bit to antifungal and also antibacterial properties of these plant compounds. And in the PKD field right now, we have sort of a little movement that is moving towards eliminating plants completely from the diet, which is the carnivore movement. However, I feel that it's more of a fad because they don't really provide any theory behind why they're doing it. It's just it's cool right now. But I think that plants are so important, and I've elucidated other points of why they're important, which is the pH and the fiber. But I think this aspect of phytochemicals that you mentioned in your book should be elucidated in this podcast as well. And you also talk about that in a supplement form. You mentioned this one supplement that's like a combination of all these phytochemicals.
Salvestrols.
Yeah, had this study which had a 100% success rate in removing cancer. So maybe you can address that just shortly, why that's important.
Yeah, the thing is, the human body is so complex and it's different for everybody. So we've got all these different people talking about different diets, what's better, carnivore, vegan. And it's difficult. It is difficult to know what the best is. So we are at this stage where there's evidence to support each one of these. But because everyone's individual, what we need to be doing is being open-minded and not jumping into one camp, because invariably it doesn't work for everybody. What you've essentially got to do is you've got to experiment. But you've got to start on a certain foundation. And the foundation for me is that, yes, on a carnivore diet, you can more easily get into ketosis, which then can combat pathogens and what have you. But then you've got an upregulation of, or an increased intake of iron. And iron is a double-edged sword. So iron is now being, there are calls for iron to be classed as a carcinogen because too much of it can fuel pathogens because it's like rocket fuel for them. They need it to proliferate. Mm-hmm. So there's all these nuances involved. So I would always suggest, if you're on a carnivore diet, you're not going to be getting all these polyphenols. You're not going to be getting all these beneficial compounds that are antimicrobial. So you want to be balancing that with bioavailability of nutrients to support the immune system, but also these compounds, because they're antimicrobial, you effectively, you want to be consuming these from plants because the plant is producing these they're literally antibiotic creating factories. So to go down this route of explaining the reason why it's so important is because two particular professors from De Montfort University in the UK, Dr. Jerry Potter and Dan Burke, they were looking at creating prodrugs, right? So these are drugs that go into the body and are utilized by the cell only when they enter the cell by a particular enzyme. And what they found was that in cancer, I'm not sure if this is the case in PKD, you might want to check this, but in cancer, there's an upregulation of the CYP1B1 enzyme. And that's an enzyme that converts, and then thousands of these enzymes that convert compounds when they come into the cell. So they knew that this enzyme is upregulated just in tumor and cancer tissue, which is very interesting. Why would that be upregulated? And because a lot of scientists think that cells have malfunctioned, they've got a mind of their own, they see this upregulation of this enzyme as a cause of cancer, so they're trying to create drugs to block it. So the scientists were looking at prodrugs that could interact with this enzyme. And these particular scientists had done work on plants before. And they realized that some of the drugs they were trying to create, the molecular structure was similar to the molecular structure of certain antimicrobial compounds found in plants. That's what led them to go, you know what, well, let's have a look at some plants and let's see if those particular plants with those molecular structures, those compounds with those similar molecular structures are converted by that enzyme only active in tumors, and if that has a beneficial effect. And lo and behold, they did. They found a huge number of compounds like resveratrol in grapes, in red grapes, that are converted by this enzyme. This actually identifies that there's a key mechanism within our cells where our cells have evolved to utilize or create an enzyme that can then utilize the antibiotic compounds in food, providing you're consuming organic food. I think this is where the problem lies. Mm-hmm. People talk about eating plants and what have you, and it didn't help them. A lot of people don't consume organic. There's a huge difference between the antimicrobial compounds in organic compared to conventional food because they're built with pesticides, right? That's a huge, that's a whole other debate.
It's because the plants don't have to defend themselves because the chemicals are doing it for them. They're getting lazy, right?
Exactly. That's a great way of putting it. So they're not producing the chemicals, so you're not actually getting access to the chemicals. So that CYP1B1 enzyme isn't being delivered any of these particular enzymes that your body can utilize. So that's really important because there's a lot of people that say that food isn't medicine, and it clearly is. I mean, Hippocrates identified that a long time ago. It clearly is, but now there's an identified mechanism of what actually happens. So our cells have created this enzyme to be able to utilize these compounds. And what's interesting is that when you look at the pathway that this enzyme forms part of, it's an antimicrobial pathway. It's an antifungal pathway. So when there's an antifungal response, certain other features of the pathway are enacted, including this enzyme. So it's almost like you could say the cell intelligently, or the body has intelligently incorporated this enzyme that also utilizes antimicrobial compounds when we consume them into an already developed antifungal pathway where the cell is able, through other means, other processes, to combat the pathogen. So there's two processes of combating pathogen here. The innate pathway within the cell itself to upregulate proteins and what have you, and inflammatory markers, and then the use of this same pathway using the food that we eat. So that's why it's so important. And so these guys basically, they renamed the particular molecules they discovered from plants as salvestrol. In a case study of about 15 patients, they cured all 15.
Amazing. And they just gave them as supplements, right? Or in the context of an organic diet, I guess?
Yeah. Yeah, because these are concentrated compounds from plants, basically.
And they're over the counter, right? People can just get them.
Yeah, they can. But they've had a real big problem trying to get funding in order to do the clinical trials that would prove them effective. Yeah. And this has been the same thing with natural compounds, obviously.
So there's a probability that they would also have an effect in PKD, right?
If we're looking at the same issue, the pathogens playing a potentially leading role. I mean, there is the point that, yes, cells do get damaged, so you're going to have an issue through the process of a cyst being formed and a battle between the immune system, the cell, and the pathogen, where you are going to get damage occurring. There might be specific damage occurring to the cell. So in that sense, the pathogen has instigated the cyst, but then it's persistent because of the damage. So there is always that element. It's not always just that the pathogen is going to be everything and then kill the pathogen.
We already have the cyst fluid, which is cystogenic in itself. It's already there. But if we stop growing, or if we, manage to shrink by 10% and then it stays like that, then we're already golden, right?
Yeah.
And we could always get like cyst fluid removed in surgery, which is available. And as soon as we are able to stop the actual trigger, that might be a great way to go, actually, because you're taking out the trash and then you don't get any new trash. So that's a good idea.
Absolutely. So just to go back to the steps, basically, that I would enact, it is address the terrain, address the microbiome, the immune system, address abnormal metabolism. But then the final thing I'm adding on to the cancer debate, and you might want to consider, is address the fungal pathogens. So we're testing in and around the tissue what pathogens are present, so we know what antifungal drugs or other antifungal compounds and the natural compounds in plants we can use that are going to be effective against them. So that's the additional aspects I'm adding to the conversation.
Going in, like biopsy and doing fungal testing?
I mean, again, this is nuanced. I mean, the trouble that we're finding with the scientists that I'm working with is the conversation around, do you do a biopsy, and which can effectively cause cells to migrate.
Yeah.
But then if you don't do the biopsy, how are you going to know what fungal pathogens are within the cell?
Right.
Within the tumor. Is it better though to just take samples from around the tumor, or take urine samples or blood samples to see if toxins are being produced? We're trying to work out exactly what the best route is, but just I suppose a basic blood test or urine test, where you can identify if you are overloaded with fungal toxins or bacterial toxins within the blood, or the presence of DNA, microbial DNA.
For example, like there's the mycotox profile that checks for, I think, 15 different fungal toxins in urine. I've actually had that done, and I have Aspergillus.
Wow, there you go. But then that's only 15. There's at least 800 pathogens that we know about.
Yeah. So how are we going to test for all of those?
But then the other interesting thing, we only have actually identified between the figure is 1% and 5% of microorganisms on the planet. We can't culture 50% of the microorganisms in the body.
Yeah.
So even the tests that we have don't find all of them. So it's wrong to conclude that, an antifungal test or an antibacterial test hasn't found any, so that therefore means that there aren't any. Well, that's not necessarily the case. Like, for instance, just remind me, because I'm going off on a tangent here, but remind me, I'm going to come back to the repurposed drugs used and using antifungal drugs. But Ravid Straussman, when he looked at this guy, this brilliant scientist in Israel, he looked at the microbiome. He was the first one who came out and identified that the tumors have their own associated microbiome, so pathogens present.
Yeah.
But when he was looking at the sequencing, the DNA sequencing to identify them, he acknowledged that 2.3, around 92% of the DNA reads was human. Around 2.1 or 2.3, I can't remember the exact figure. It was microbial, so it was bacterial and fungal. But then there were 6%, just over 6% reads that were unknown. So they couldn't identify that DNA in the current databases. So you have this potential for all these other microorganisms that are present that we haven't yet been able to identify, and we don't know what they do and how they're affecting things. So, there's this unknown element. So we can't say concretely that just because a test doesn't find something, that they aren't present. That's the crazy thing. Yeah. That's what we need to keep in mind. But the other thing I would like to add with the fungal aspects, the additional fungal treatment I'm identifying that we should be looking at, this fungal aspect, is that when scientists are now looking quite deeply at antifungal drugs, because they're finding that they are effective or have efficacy against a broad range of cancers. Now that's interesting in and of itself. And there were several case studies. So well, there's two, like in pancreatic cancer, for instance. One particular 65-year-old pancreatic patient was diagnosed with inoperable pancreatic cancer. He was terminal, but he had an overt fungal infection. So the treating physician gave him a 9-month course of itraconazole, right. And that reduced the size of the tumor in the pancreas, allowing it to be resectable. And he was cured from this incurable disease just purely because of the drug. And the other thing is, they've done tests in pancreatic cancer where they find that Malassezia fungi, the lipophilic fungi, increased 3,000-fold within that tissue, and they're present within the tumor. And when they used particular antifungal drugs, they reduced the tumor by 40% and it stopped growing. Then they reintroduced the fungal pathogen and it started growing again. So with those experiments, there's conclusive evidence to show there's a correlation, major correlation between the progression of the disease and these pathogens being involved in that. But the other thing is, when talking about targeting cancer using an antifungal approach, when you look at all the off-label drugs that are now being touted to target metabolism and other features, other pathways within cancer that other people are using, this is the big thing, using ivermectin, using antiparasitic drugs, using metformin, even doxycycline, which is an antibacterial. When you look at all these drugs, they are also antifungal. Wow. They're secondary, they're unknown properties. They're all antifungal. So technically, I'm suggesting, well, there's this huge correlation pointing to this fungal aspect. Even the drugs that are now being used that are thought to be acting on another pathway that pathway is also antifungal by virtue of the fact that it reduces the access to the fuels that the pathogen needs, but also they're antifungal in their own right. So they're targeting the pathogen themselves in a different way. So are these drugs working because they're actually inadvertently targeting the fungal pathogen I'm saying is involved in cancer? And maybe they may also have efficacy with PKD, because it does seem like there's a pathogen element, a fungal element. But I suspect, as with the tumors, there is also this bacterial overlap and synergy of them working together.
Yeah, it sounds like we do need to find at least some common sense versions of these treatments, right? I mean, there's companies that will test your stool and then they will sort of give you recommendations on which foods will optimize your microbiome further or help with pathogenic species. And then there's these salvestrols, for example, that you're mentioning. We can do an organic diet. We can do grass-fed beef. We can do a lot of vegetables. And of course, also make sure that we don't have too many anti-nutrients. As you also talk with Dave Asprey, you know about that whole area. So we don't want to poison ourselves. But then again, we need the plant compounds.
Yeah, that's why you don't want to be completely plant-based.
Yeah, you want to have both, and then maybe steer clear of the worst offenders that have the most anti-nutrients, like grains, for example, can be an issue. But all these vegetables can be of so much value. So those could be some tenets to go by, like ketosis, optimization of the gut microbiome, salvestrols, maybe salvestral supplements. I haven't tried them yet, but it does sound like something that's very affordable and can just be added.
Yeah, like I said earlier, it is a case. Unfortunately, we're at the stage where we have an idea, we've got an inkling of what it might be, this and that. Several things are involved: metabolism, DNA mutations, blah blah blah, the microbiome. So, from a treatment perspective, it is try these things and see what the contraindications are with other drugs.
And of course, like if you can find actual fungal infections, then treat them, right? You can find fungal infections in the stool. You can find them in the urine. You can probably find them in the blood, which, of course, they're probably going to be very, very, very extreme at that point. But they need to be treated, right?
They do. But I want to touch on another important point, and that is, well, 2 points really. Just because you will use a broad spectrum antifungal drug like, say, itraconazole, doesn't mean that the fungal pathogen that's present doesn't have resistance to that drug already. So that's another nuance. So don't be despondent if something doesn't work. Try something different and be mindful of not using monotherapies. You want to really be targeting this from several different points of view, because we don't know exactly what combination of pathogens are involved. The other thing is, say for instance, itraconazole, it's fungistatic. It's not fungicidal, right? So a lot of drugs, fungus are do not eliminate necessarily the pathogens, they just inhibit them. So this is where the immune system is so important. You've got to really optimize the immune system, ensure that it's working at its full capacity, because there were studies done by in AIDS patients where there was a fungal infection, say, and the treating physician piled in as many antifungal drugs, high strength as they could, that they knew could kill the strain of pathogen that was there. But because the immune system was shot, it wasn't able to eliminate the fungal infection. So it just highlights that there's this dual combination. The immune system is the army, essentially, and the drugs that we use is the air support. You're bombing down or whatever it is you're using to inhibit the opposition, which then allows the army to go in and take them out a lot easier. But you need the immune system, and specifically with cancer, there's an issue with glutamine. So mentioned it earlier, and you were talking about glutamine to me earlier. With any chronic condition, I mean, just to preface that, sorry, our muscles produce 90% of the glutamine our body uses. So glutamine is a non-essential amino acid because it's so abundant, right? But when we're in a chronically inflamed state, proliferative cells such as the immune system need glutamine to function. They use it, and they use it up a lot. And when you go past a certain level of chronic inflammation and maybe toxin exposure within the body, the immune system becomes depleted, and the ability of our muscles to produce enough glutamine is diminished. So glutamine then becomes a semi-essential nutrient, and this is not often addressed, which means that if the body's not producing enough glutamine, the immune system, although there might be a sufficient immune cell count, the immune cells themselves are not working at their optimum because they can't get access to the amount of glutamine they need because it's lower than it actually needs to be. So this is where there's controversy. This is a controversial area now because I'm pondering on the side of supplementation with glutamine with cancer patients, and so this will depend on whether or not the cancer is highly glycolytic or consumes a high rate of glutamine itself. So there's nuances in this, but the bulk of evidence suggests that supplementation of glutamine is actually more beneficial because the cell itself is limited, it's rate limited to the amount of glutamine it is absorbing. And it's always got access to that glutamine anyway because the body produces so much of it. Yeah. And the issue is that the immune system is actually reduced, and the gut is reduced. Permeability of the gut is increased because you haven't got the access to glutamine. So if you increase it, what you're actually doing, you're not actually increasing the amount of glutamine that's entering into the tumor, but you are increasing the ability of the immune system to see the tumor.
Yeah, the listener can sort of think it as like you're leveling the playing field because the tumor is already getting dibs on the glutamine anyway, and it's going to have enough. And the rest of the body is going to like give some of its glutamine to the tumor cell. So now the tumor cell has more glutamine than the rest of the body, which also needs it. So if we supplement glutamine, we have an improvement in the gut lining, an improvement in the immune system. And to round that off for listeners that have listened to the episode with Professor Seyfried, I think it can be combined. So we can make sure that glutamine is sufficient in all the tissues, and we give the tumor the glutamine access anyway. And then we could still, if we have a proper strategy, which most people don't, we could still go in and for an hour every couple of days block glutamine with something like Dawn and see a good effect because you're leveling the playing field in that case as well, because nobody gets access to glutamine or everybody's going to get access to glutamine. So it works actually together, I think.
Yeah, I do think there's definitely possible, but there's definitely scope with that. Absolutely. I think the only downside, obviously, to restricting glutamine would be the restriction of the immune system. But as you say, if you have that in mind, then you are going to increase and supplement the glutamine as well to then bolster the immune system in that process, whilst you're weakening PKD and the tumor as well in terms of cancer.
It's a very interesting combination.
Yeah. I think we need to see, I think Don's difficult to get hold of, isn't it? So we need to see Don being used in patients and what the side effects are. You suggest just an hour's use, that might be enough.
And I'll work on getting somebody who's doing the therapy with patients and the Don on the podcast in the future. I already know who he's going to be, but I have to get a hold of him. But that's going to be interesting as well to see how they do it. And I'll ask him to consider that glutamine supplementation actually on the other end. Yeah. Wow. Okay. I think that was a great wrap up. Also to have some actionable advice at the end there, and that'll give people a lot to think about in regards of organic vegetables and maybe getting checked for whatever fungal infection might be there. I might link some of the testing down below for things that can be done. So where can everybody find you and your theory online for further reading, and your great book, of course?
Okay, so I've got a website which is full of abundant information. I've also got a free PDF on the website, a synopsis of my theory, so you don't have to read the entire book to understand the basic principle. The website is cellsuppression.com, and my book is called The Cancer Resolution and is available on Amazon. You can get that just by going straight to Amazon or the link on my website. So yeah, they're the two main places. I do have a Facebook discussion group where patients come and we discuss the theory and the implications of that for treatment, and everyone works together to help each other out, answer particular questions that patients have. So by all means, you can join that group too, and the link is on the website.
All right, and I'll have it down below as well. Thank you so much for coming on the podcast. That was very insightful, and I think our listeners will dig it as well.
Thank you, Felix. It's been very nice chatting to you about this. Hope to see you soon.
Wow, everybody. I think Mark's theory is so amazingly close to what I think is happening in PKD. Just that I focus on endotoxin and he focuses on fungus. Now, are those mutually exclusive? Probably not. I would not be surprised if both can be found in PKD cyst fluid. And they have been found there, of course. The question just is, do we need either, or do we need both, or do we just need one of them? And I think probably the answer might be we just need one of them. Just the studies have happened to focus more on endotoxin in the PKD area, and Mark has focused on fungus. But in Mark's case, it looks like the fungus is a necessity. In PKD, we don't really know if it's really just endotoxin or if it can be a variety of toxins that all trigger this. But we are pretty sure that endotoxin can trigger cyst growth. Check out some of the links that I have below for some of the products that we discussed if you want to try some of them. And I suggest you might want to listen to this one twice and maybe send it to a friend, because I think it has so much valuable information. You might even want to send this to friends who have cancer, because I think Mark's theory really has the most merit that I have heard for now. All of this does not go against Professor Seyfried's theory of cancer. I think that it supports it. And I think that it is like 95% aligned with it. But it does change the approach to it a little bit, since if we just address cancer, or maybe PKD, by rescuing the uncontrolled growth, but we never solve the actual issue that is in the background, which might be the pathogen or its toxins, if we're correct on this, then we will always get recurrence as soon as the therapy isn't strong enough anymore. So for example, there are cases in cancer where people get into total remission and there's basically nothing left of the cancer on a scan anymore when they do an extreme ketogenic diet for years on end, right? So they have very high ketones, much higher than standard nutritional ketosis. So for example, a GKI of 2 or below, right? There are some of these cases that get to basically zero cancer on a scan. And then they go to a GKI of 4-1 or 5-1, which still is where I am basically most of the time, right? I'm not doing extreme ketosis. I'm doing long-term nutritional ketosis, right? And in some of these cases, the cancer comes back with a vengeance. And even though people are still in 4-1 or 5-1 GKI, they die, right? So if that is true, then it's very unlikely that it's enough to just address the mitochondrial problem by giving them a nudge with ketosis alone, right? It doesn't seem to be enough in cancer. It just seems to be enough to keep it at bay. But this to me means that the tumor itself is a protective mechanism by the body, just as the cyst seems to be. And if we just focus on suppressing that protective mechanism, suppressing the cyst growth, suppressing the tumor growth. This might be enough to keep it at bay, but as soon as we let off a little bit, it seems to come back with a vengeance. And I think that looks very much like if you stop treating a wound with antibiotics too soon. The pathogen comes back. So what if instead we do focus on mitochondrial function, improving it, getting into ketosis, all these things that we're already doing, also in PKD, but many cancer patients are doing. But then we combine it with our own approach against the supposed pathogen, using salvestrols that we talked about in this podcast to increase the body's resilience to these fungi and also to re-enable the apoptosis of damaged and infected cells. What if we went the route of doing some gut testing and start eliminating some of the pathogenic fungi that are probably in our gut and even some of the endotoxin producing bacteria? Could we maybe find some approach where this kind of pathogen is eliminated or suppressed to the degree where the immune system can finally totally get rid of it? We don't know the answer to this question, but the existence of these cases in cancer where people almost get it to zero and then it comes back with a vengeance just because they let off on the protocol a little bit shows to me that there has to be more to it than just mitochondrial dysfunction. It has to be some kind of pathogen, toxin, some kind of event that is offsetting it in there, even when you've almost gotten rid of it. So this is just some food for thought for you guys. I don't have the solution for this yet, but we'll continue reading research papers and probably try and push some of our own ideas into the research field. I'll see you on the next episode. Until then, happy healing.